So for our next speaker, we have Chris Rackauckas from the Massachusetts Institute of Technology, from the University of Maryland, Baltimore, from JuliaComputing, and nonfocus, if I'm not mistaken. And he'll be talking to us about Pumas, the pharmaceutical modeling and simulation engine.
All right, hello everyone. Viral actually gave a very small introduction yesterday at the Julia 1.0 talk. Where we're going with Julia now is not just solving these mathematical problems, but real world problems. And he mentioned that there is something that we're doing with personalized medicine, and that is Pumas, this pharmaceutical modeling and simulation engine. And we have these major affiliations, and that's one of those things that we're going to have to explain. So, okay, what is this? Well, this project was started by the Center for Translational Medicine at the School of Pharmacy in University of Maryland, Baltimore. So if you don't know, translational medicine is the process of going from medical research inside of a laboratory to medical research that's actually used in clinical practice. And so this group was interested in trying to see if we can use computational methods to be able to improve the translation of these theoretical ideas or these things that we're trying in a lab and actually bring them to actual practice, like bring them to become actual medicine that we're using in the field. And there was actually a review article that came out, I think this was last night, which was that everyone sees that the future of clinical trials needs to be able to use these ideas, these AI, big data, technology, to be able to make drug development faster, cheaper, and more effective. And how can we do that, right? Well, the reason is because these days we have all this data on given individuals. We have your electronic health records, and we know every single time that you've been to the doctor, we know every single reading about you, and we know what kinds of diseases you tend to get, and we tend to know how you respond to drugs differently. And so the question is, can we use that to be able to computationally come up with better methods to explore drug development and explore the kinds of drugs that you should be getting? And so there's a comprehensive push towards modernization, right? There's billions of dollars that goes into drug development every single year, but we're still using a lot of the same old methods, which is, you get a cohort of 30 to 50 people, and you try a drug on parts of them, you randomize the trial, you give some people a placebo, and then maybe you didn't get a certain amount of, maybe you find out halfway through the trial that being overweight or being of a different ethnicity actually affects the drug. And you might need to go all the way back and have to get a different cohort in order to do this trial effectively. Or maybe you did not have enough individuals in your trial to actually get statistical significance. These are the kinds of things that we can try to see beforehand, right? We can try to see through clinical trial estimation and simulation what we need to know about the cohort in order to actually actively guide the clinical trial such that we can more effectively get to the end goal, which is a new medicine which is approved by federal agencies like the FDA, so that way we can treat real people in the real environment. And so bridging the computational approaches of drug development to basically the way that we're going to the future, it involves many different techniques. And so we can, in some cases, entirely eliminate clinical trials by doing certain estimations where we can estimate how, if we have one drug, which is a lot like another drug, or if we have one pill capsule, which is a lot like another pill capsule, if we can understand how it releases that drug differently in a lab, and we can understand how people respond to that drug through clinical trials, maybe we can mathematically understand whether this new formulation of the drug is safe without having to rerun entire clinical trials. So these are the kind of things that we're looking at for can mathematical simulation actually affect the process through which drugs can be approved and be put to use. And so what happens when medicine enters the body? Well, it is dependent on the type of individual that takes the drug, right? So here it shows that there's a different profile for the concentration of the drug inside the body for males and females. But it's not just males and females. There's so many other factors which we have to take into account here. So if you take many different individuals, you'll see that each of them will have a different drug profile. They'll have different concentrations over time. And these are the kinds of things that we will want to be able to account for. These are the kinds of things that we will want to be able to identify. So from very simple information that's easy to measure about an individual, can we find out which of these drug profiles you will have? Can we find out exactly how your body will be different from somebody else's in the way that it's able to process the drug? So If you thought, well, it's Chris talking and there might be ordinary differential equations or something involved, you were correct. Because the way that these are modeled is through ODEs. So here's a very simple ODE. This is called the one compartment model. And so we can have these very simple ODEs which say, here the drug enters linearly and then it goes to a central compartment and it decays off from there. And the interesting thing about these models is that the way that this might describe how the drug is in the body, but we don't exactly know the rate at which the drug decays in your body. When you come into the clinical trial or when you come to the doctor's office, what we are actually able to measure are things like your weight, your height, your sex, your ethnicity. And what we need to do is we need to be able to infer how your internal differential equations, how your internal drug concentrations are changing given the values that we can measure. And so what we can do is we can create these models which relates these real covariates, these like weight and sex, which we can measure, to parameters in the differential equation, to a differential equation that we can actually solve and estimate how you are doing. And then we can repeat this process on entire groups of entire populations and be able to better make this model understand how your body will be affected by these drugs. So this is what Pumas.jl does, or Pumas.jl. It is a pharmaceutical modeling and simulation engine which is written in Julia. So it does these simple models like PKPD models, but also does more advanced models as well. So there are things known as physiologically based pharmacokinetics models. These are models where we might model your entire heart, and we might know entire relations between covariates to parameters in how your heart and your liver and your bloodstream responds to a given drug. And then we can model this entire system to be able to get more accurate measurements of how you will be affected by the drug. And even larger models, such as quantitative systems pharmacology models, are able to be also prescribed in these ODEs, DAEs, DDEs, SDEs. All these different types of differential equations are the way that these models are described. And what this simulator is able to do is able to build that link between the covariates and the real differential equations, solve these differential equations, and be able to create these simulations of entire populations, and be able to understand how different people will be affected, and be able to understand what would, be able to start to try to predict the right way to dose different given individuals, given the properties that we can measure about them. And so why is this the software something that's new and different? Well, it's built using the Julia language, which it's able to get performance. And so this is something that really matters when we're trying to scale this up to very large models and large populations if we want to solve them all at the same time. So Vero mentioned the other day that we're trying to be able to compile an entire ODE solver directly to a single GPU kernel so we can run them thousands and thousands at a time. So that way we can do entire parameter searches over full populations. And that is, this is something that's part of this Pumas.jl project. And so we can use Julia macros to specify everything in a high-level language. So that way we have a low-level interface. So that way all of our mathematicians can play around with making the most intense models. But at the high level, clinicians can actually use the software without having to spend the time to really learn Julia and be able to use this within the clinical trials. So we will actually be having a clinical trial starting with Johns Hopkins in January, where we'll be putting these techniques to use. And so it's going to be open source. And open source in this case means open validation, right? So since this is going to be something that's going to be used by the medical field, we want to make sure that everybody can see exactly what our tests are. And so that way everybody can know what we have validated, what we have not validated, and it's something, and so that way other people can access our validations and can add more tests as needed. So that way we can know that we are correct, right? The problem with the closed source software, as you might have heard Tim Holy say the other day, you can report a bug 10 times, one time a year, and it might just never get fixed. But here you can see all of our code, and you can know exactly what we've tested, and you can always you can join in on the conversation and let us know how to make this more accurate, because that is something that is required in medicine, because we're dealing with real people. We need to ensure safety. And pervasive automatic differentiation and symbolic manipulation is something that we have in Julia, and this is really required to be able to do parameter estimation well. So because we want to not just be able to simulate, but we also want to be able to, in the future, be able to find out how to exactly build these bridges between the input data and be able to find out what that means about the parameters in our model. We integrate with the data science tooling of Julia, which is developing well, if you've seen the query reverse talk. If you haven't seen the query reverse talk, go find it on Google or on YouTube, same thing. And it's built on top of differential equations.jl, and I will show you exactly what that means about how the software is able to do very complex modeling. So and before saying anything else, let's just simulate something. So let's declare some parameters and covariates. Here we say that there is some parameters, sigma, or theta, and it's a vector of 4 different parameters. And then we have a positive semi-definite matrix, and we have some sigma, which is going to be a random noise. And we can say that we have these etas, which are random factors. So this would be something that is different between individuals in a population. And we say that this goes to some normal distribution, which is positive, based off of this matrix. And we get from the data each person's sex and weight. And now we can define how we go from our data to our ODEs. So we say that our ODEs are defined here. You see that that matches the differential equations.jl syntax, where we have these parameters Ka, Cl, and V. And how they're defined is by these input parameters, the theta, and also our data, which is the weight and sex, which can be different for each individual. And then when we define an error model, which says that what we're interested in is the concentration, which is the result of the central compartment divided by a person's velocity, or not velocity, the volume of that individual's gut. And then if we add a little bit of noise to it, this is a model of, okay, this per individual has this input, this ODE, and from that ODE, we're able to measure a certain quantity from it with a little bit of noise. And if we do this, what kinds of things are we looking at? Well, so here I'm just showing that, here we actually pulled from a data set, given covariate, or these sex, weight, and other features. We put in some parameters, and then when we simulate it, we are able to see what an entire population is going to look like for this type of ODE model. And so what we do is, in this case, we give them one dose, and then from that one dose, the drug enters the central compartment, and then we're able to see how the parameters of each individual makes the drug within their gut be able to behave differently. And so there are many dosage regimes that we can that are available. So there are previous programs that have been able to, such as something called NLME, an old Fortran 77 software that is like the industry standard for what are the different types of doses that you can be done in a clinical trial. And what we've done is we've shown that, so if we have like a steady state dose, which is someone keeps on going to the clinic, and after a given time, their body will adjust to having this constant dosing rate. Can we find out what that steady state will be and instantly get them to that steady state as though they've been in this clinical trial for many months? And if you look at our results against the previous software results, we're able to pretty much match it exactly. It's not exact, it's only to 12 decimal places, but I mean, you can see that in our validation. We don't just have it in one case, we have it in pretty much any case that non-mem that you're interested in. We have been able to validate that we can at least match their simulations on basic ODEs. So what can we do though, beyond basic ODEs? Well, we can go to various implementations of these larger models, right? So one of our GSOC students actually took a look at this model, which is this is something that we call this physiologically based PK model, where now we're modeling we're basically using a lot more biological knowledge about how this drug works within the body. And then we write this gigantic ODE, which I can't even show the ODE because, oh, there it is. Yeah. How the gut, the kidney, the liver, and all these different pieces work together, and then we're able to directly simulate those kinds of models. And we're able to verify this, once again, with open tests, because we have this open source software to be able to do this. And that's something that's different from a lot of the previous incarnations of this kind of work. And so this is actually a joint project that's being done by University of Maryland in Baltimore, in the School of Pharmacy, and MIT and Julia Computing. These three groups have come together, and we're going to be applying for a center for predictive healthcare analytics, which is going to be based around this software and its use within the clinical trials. But I want to answer one question before we really finish up, and it's, here I am someone from the MIT mathematics department, and, differential equations developer. What does bringing our type of community into the healthcare community, like what are we actually able to give to this community that's different? And well, a lot of these things are these very Julia things that we really want to share, right? So we can have a bunch of unique methods for ODEs. We can have GPU acceleration, and I think this is best shown by pictures, right? So the industry standard for a very long time was to use these integrators like the gear integrators, the LSODE or CVODE. And a lot of people have just, they have shown to be the best in a lot of test cases for your standard ODE solving. And so they were applied to this domain. And so if we just look at one ODE with one event and we say, how well do these methods do in this case? Well, what I can show you is that they don't do very well in this case. And the reason is because whenever you have an event, the standard integrator for these stiff ODEs, what they need to do is whenever you have this discontinuity, they actually need to start back at order 0, reduce their time steps, and all sorts of adverse effects occur. But what we're able to do with differential equations.jl is utilize methods which are specific to this domain. So we can, in this case, it only needs a step directly to each of the events, and we can have an interpolation fill in what the intermediate values will be at an even higher accuracy. So that way we can increase the simulation speed while also increasing its accuracy, and so we can scale this up to larger models and larger populations, because these are the kinds of adverse effects that, when people mostly tested these methods, if they weren't testing it on a model which is all about dosing and events, then they would not have seen this kind of effect. So the main thing is that Pumas can use any type of differential equation solver in differential equations.jl. So these two projects can grow together, and as one increases its efficiency, the other will as well. So in addition to using new methods, we're also able to make use of GPU specialization or acceleration. So I have a whole blog post showing how we've been able to make differential equations.jl's solvers compile in such a way that they can make use of arrays on the GPU, and we can solve stochastic partial differential equations using GPU methods. And here's just a little quick look at it. I'm not going to go into detail because you could read it more. But the idea is that using Julia's broadcast mechanisms and Julia's array types, we're able to compile entire codes to the GPU. And this is one way, once again, where we can scale up to these larger scale models and be able to get more accurate predictions. And we're also able to utilize these models which incorporate different types of stochasticity, right? So we might have an ODE model of the drug concentrations within someone's body, and then one of the ways that we usually talk about pain is we talk about pain as a scale of, what's your pain, 1 to 9, and it's always a discrete number. Can we actually bring discreteness and stochastic, and differential equations together? This is one of those things that we've been able to demonstrate we can do, where you can have these discrete numbers mixed with these continuous numbers in one given simulation. And so we can bring together models that were, previously were simulated very differently, or you had to scale down the type of model, but now we can bring them all together to be able to, once again, be able to just make more accurate predictions by using the full-scale models of how we believe humans are interacting with the drug. So continuous solutions do give us more accurate diagnostics, right? You might have heard that differential equations.jl was all built around continuous solutions. And so what we've been able to see is that that's able to give you much more accurate answers. So here's actually an example where here's a harmonic oscillator, and we might ask the question, can we find the minimum and the maximum of the solution? Right? So you might have a drug inside the body, which is, if you keep on giving it different doses, it's going up and down. If you had 50 points, would you have gotten a good test of what the concentration maximum and minimums are from that picture? And so if you just took 50 points out of there, you might say, well, I might just take what the minimum and the maximum are from the points that I've gotten from my differential equations solver. But from using differential equations.jl, we can say, well, we have a continuous solution, so let's throw this on a global optimizer and actually find out what the true maximum and the true minimum are. And you can see that they can be very different values, right? If you took 50 points, you might have thought you got a good solution, but instead you were completely wrong on where the time was that the minimum solution was achieved, and you were completely wrong on what it was. And the reason why this is interesting in this case is because when we were trying to find out whether a drug dose is safe, what some of these diagnostics are is, what is the maximum concentration that is achieved during this trial, and what is the maximum, or what is the area under the curve, right, the integral. And by using these interpolating polynomials and these continuous solutions, we're able to get very accurate solutions on what those values would have been. And so once again, we're able to more accurately be able to apply these models and these methods to be able to understand how humans are being affected by these drugs. And once again, be able to personalize how we're doing these dosings by these predictive analytics, where we're predicting how each person how each person is going to have a very different reaction to the drug. And one nice thing that we can do about this too, a very Julian thing, is that we can propagate uncertainties all the way through our differential equation solvers, right? So we might say that we have some uncertainties in the input parameters. What does that actually mean about the uncertainties in the outputs? One traditional way of doing this is you'd say, well, if I have some uncertainties, let's sample some possible parameters and run the differential equation solver thousands of times. That's something that's been done a lot in this field, but it's computationally intensive. But instead what we can do is we can use the type system to be able to represent numbers we can represent numbers with uncertainties, right? So numbers as probability distributions, as these Julia number types, and be able to directly put it through our simulation methods such that we say, here's uncertainty in our inputs and our parameters. What does that mean as the uncertainty in the outputs? And then we directly get the error bars in our output solutions. And this really matters because whenever you're dealing with healthcare, right, you need error bars in any of your things to be able to tell you how safe it is. And so we have direct ways to be able to calculate uncertainties and propagate them through entire models in an efficient manner. So that way we can scale up our models, but not lose this idea of uncertainty. And another thing along the same lines is global sensitivity analysis, right? So if you were to change given input parameters, what about the outputs will change? And what we're able to do is we're able to show, this is actually on that large scale, the PBPK model that I showed. We're able to do global sensitivity analysis, which basically tells you what do changes of input parameters, what do they correspond to in the output? So what this is showing right here is that the concentration maximums, if you would have changed the rho parameter, you would have greatly changed the concentration maximum, whereas changing some of these other parameters would not have done very much. And by being able to do these sensitivity analyses on these full models, what we're able to do is we're able to direct the clinical trials and basically tell them, like, hey, these are the parameters you need to be researching, these are the parameters you need to be measuring, so that way we can get better and better predictions as we go along. And so the nice thing is we have to put everything together, right? As the models get larger, if we want to do global sensitivity analysis, we need to solve it thousands of times, and we need to be able to solve these large models thousands of times, and so we need to have everything be more efficient. And so that means we need to be able to put these types through, get the uncertainties out, we need to be able to put things on the GPU, and it all comes together as one story, which is, big models, solve fast for more accuracy. That's the general idea. And so this is how we're all working together, right? Julia Computing and MIT, we're improving the computation and the mathematics to be able to scale up to, something that is just a more predictive model, just by, increasing the computational power that's involved and the mathematics that we're using, and then we're directly applying it to real clinical trial situations through our association with University of Maryland, Baltimore. So this is a recap of where we're at. The simulator has actually been tested on these types of models, these smaller PKPD models, and then these larger PBPK and these QSP models. We have the DSL that I showed, though it will be changed a little bit before we release it. We do have automatic and symbolic differentiation for helping with getting derivatives of these solvers and being able to perform parameter estimations. We have continuous outputs, and we have new high-performance solvers, which were created for these specific types of models that are seen in these PBPK and QSP cases. We have these dosing regimes which are implemented, so that way we can utilize different clinical trial situations. So we have these, these models where then we're able to affect the individual with different doses, and with steady state dosing and other continuous dosing or IVs. We can model all these different situations, and we've been able to verify those situations against the previous software. And we also have all sorts of new GPU acceleration and new integrators which are being used to be able to improve our performance. So this is all one giant project where we're going to be not only do simulation, the simulator, this is just the first thing that we're showing. The other parts will be bioequivalence studies, so we can show how whether drugs are equivalent enough to be able to pass the FDA regulations. And we're also going to be doing things known as IV-IVC analysis, so in vitro in vivo correlation studies, and optimal experimental design. So basically we can use this software to be able to say exactly how experimental designs can be changed so that we can optimally dose in the future and iteratively increase this. So we're going to actually have this made in a web app format, and it's going to be put to use once again in the clinical trial starting January by Julia Computing, and so the app is being created by Julia Computing. Some of the mathematics is being done by us at MIT, and it's all being put to use by us in the University of Maryland, Baltimore for this, translational medicine. Because this is all about bringing this is translational medicine and translational mathematics, right? We're bringing new tools to be able to bring we're basically to bridge from the research to creating new medicine through these mathematical methods. Thank you very much.
Thank you very much for your talk. Does anyone have any questions?
Thanks, Chris. That was really amazing. So I have two questions, if I may. The first is about this kind of metaprogramming interface for problem definition. Could you please go to this slide back again? I actually did not really get anything, but you also did not say anything. So I'd like you to say a couple of things. Because from what I saw, it wasn't really that much different or more usable than the already existing standard problem definition. So yeah, this example or the previous one even better.
Yeah, so what it's actually doing in the background is, so there's a complicated callback. That's really the issue here, right? So we can define these differential equations, but the differential equation on its own is not useful for this domain because you also have to be able to specify exactly the clinical trial schedule, right? How they're getting doses, what kinds of doses they're getting, and really what this is doing is this DSL is letting you specify like here's the model that we want to solve. But also in a very clinical trial manner, you just say, and I want it to be dosed in such a way that, like this is an IV bolus dose for this duration, and it's able to internally build the callback to be able to then solve that equation properly. So the DSL is really to a higher level DSL to make it so that way it's easier to do these full scale models with the events, because the events are the difficult part here.
All right. Okay. So I also have one more. Could you tell me what is the performance of using measurements.jl in solving? Like, for example, if you solve one with measurements instead of just float 64, what's the ratio of time it takes?
That we don't exactly know yet. But what we do know is that it's much faster. It's not orders of magnitude slower. And so it's still much faster than solving the ODE 100 times. And there's still different ways that we can still improve the performance of the measurements.jl. And we are talking with the authors there, and this is ongoing research on that part.
All right, that's excellent. That's excellent.
Do we have any more questions?
I'm going to be greedy and make 2 points. The first thing is that you mentioned the queryverse. I went to the queryverse tutorial on Monday. And it still blows my mind that you can link up differential equations with the queryverse to what seem to be completely different things, and they just work seamlessly. I am amazed.
Well, thank you. Well, I mean, yeah, it's this project, right? We want to be able to simulate, but in the end, we want to get CSV, so that way, someone who's not a Julia programmer can, look at what the predicted concentrations are in a manner that they know. And David Anthoff really helped me get that done.
During your talk, you said, what value can mathematicians and programmers like us bring to the medical field? You can bring a lot, you know that. This is nothing new. When I was a bit younger than you are, in fact, when I was in high school, I learned to program in Fortran on a DEC PDP-11, which is probably primitive these days. That machine was in a research unit called the Diagnostic Methodology Research Unit in Scotland. That unit was set up by a man called Professor Wilfred Card, and in 1967 he produced a paper called A New Calculus of Medicine. So this field is you know, it's been going for 50 years. I think it was one of the seminal papers in this field. I'd love to get you a copy of it, actually. I think
Okay.
you'd be very interested in it.
Yeah, we could talk after this. Yeah.
Does anyone else have any questions or?
Torkel?
Okay. Then I also want to ask one last question and we'll need to end the session.
How much does the type of drug impact these methods? Can you like apply it to like a very wide range of drugs, or is it like you're limited in?
Well, what we're limited in is the to the types of drugs that we actually fit these models to, right? So when we create these models, we also have to be able to use some form of clinical data or electronic healthcare data to be able to actually fit the models, find out the relations between covariates and all these. The parameter fitting thing is not something that is completely done, so I haven't shown it here, but that is one of the things that this is an ongoing project where we've hired now multiple developers throughout the Julia community, and we through this funding we are building the more and more of these nonlinear mixed effects models, things to be able to fit these parameters in both optimization and Bayesian methods. And we're also looking into using machine learning techniques to be able to mine the electronic health records data, which is not just text, but it's in natural language processing plus image processing problem to be able to then get these parameters. But if we have sufficient models and if we can get sufficient parameters, then we can run these simulations and be able to do all this. And so yeah, each drug might have different parameters, but as long as we have enough health data and as long as we have something to match it to, then we can start being able to use these methods. So yeah, there's a lot of different ways that we're exploring that, these machine learning mixtures with differential equations. These are the kinds of things that are new that we're investigating behind the scenes with this project that we'll be releasing. So this entire thing is actually under the Quant Farm Tools organization, and this is just the first part that we're releasing out of this.
Okay. Unfortunately, we're over time, so if we could thank the speaker again.